Next up, we have Dr. Olufemi Alabi, who will be talking about squash leaf curl virus.
So thank you very much for this opportunity. I'll be talking about squash leaf curl virus, but a lot of what I'll be talking about are probably things you've heard from other presenters. These whitefly-transmitted viruses share a lot of commonality in terms of the way they move around. And so, pardon me if some of those amount to repetition, but hopefully they serve to kind of emphasize, some of these important points. So squash leaf curl disease was first observed in winter and summer squash in about the late 1970s in the US, but specifically in California and Arizona, but also in northern Mexico. It infects mostly plants in the Cucurbitaceae family. That will include cultivated cucurbits like watermelon, squashes, melon, cucumber, and pumpkins. There are differences in the way these different cucurbits respond to squash leaf curl virus infection. Losses are more severe in pumpkins, squashes, and watermelon, and they are typically minor in cucumbers and melon. Once shortly after it was discovered, observing the field, the causative agent was identified to be squash leaf curl virus, which is a begomovirus, similar to some of the ones we've heard about, and it's a new world bipartite begomovirus. Sudeep during this presentation showed you an electron micrograph of what the virus particles look like. Is going to be similar to what squash leaf curl virus particles look like. And just like the virus that Cucurbit Sudeep presented on, this is also bipartite virus in that it has two DNA copies, each of them about 2.6 kb in size. As you can see, the DNA here is where a lot of the genes are encoded, mostly for protecting the genome, but also for replication. But the DNAB also encodes some genes that allow for systemic, movement of the virus within the plant. Multiple strains of squash leaf curl virus have been characterized. And the differences between these different strains is, their biological properties. There are some strains, that have a very narrow host range even within cucurbits, and some strains are able to infect, a wide range of cucurbits, but also some non-cucurbit crops. And similar to other begomoviruses, squash leaf curl virus is also vectored by Bemisia tabaci, the B biotype, or the silver leaf whiteflies, or MIAM1 as it's called now, particularly in the US and Mexico. And like you've heard from Bill, in some growing areas, like in South Texas here, whitefly populations can be very, very abundant, and that drives the epidemics of squash leaf curl. In terms of symptoms, multiple symptom types have been recorded for squash leaf curl. They consist of mosaic or mottle, which can be either bright yellow or mild, leaf curling or ination under the leaf, which makes the leaf have this distorted appearance. When infection occurs very early during the season, the plant can become stunted. The flowers will be late in opening or even prematurely abort. And the few retained flowers, may not set very good fruit, or even the fruits that are set may have delayed maturity or become malformed. It's important to emphasize here that symptoms may be very tricky to interpret because symptoms may differ between different cucurbits. The timing of infection may impact the severity of symptoms, but more importantly also because mixed infection status of plants can make symptoms very confusing. Here's a picture of a symptomatic plant, presumably from Jordan. It is unclear whether this is only due to squash leaf curl virus or whether other viruses are tested. And so we have to be very careful in the way we interpret symptoms. But in our own growing condition here, we've been recording different symptom types on different cucurbits. Here are symptoms on young pumpkin leaf and matured pumpkin leaf as recorded due to single infection of squash leaf curl virus, which I must emphasize this is very rare to come by, but once in a while we do have situations where we have single infections. These are different leaves, singly, but also mixed infected of squashes, zucchini squash in particular. And you can see, as you can see, it's really difficult to tell based on symptom, whether a plant is singly infected or doubly infected. In this case, mixed infection of squash leaf curl with CYSDB that Bill presented on previously. Here's a watermelon plant that has chronic infection of 3 different viruses. CYSDV that Bill presented on, SQVYV that Scott presented on, but also squash leaf curl viruses in the mix. And so it's really difficult to tell what exactly, is the symptom due to one or a combination of these? Are these viruses acting synergistically with each other to cause more severe infection? And just like you've heard before, some wild cucurbits, like buffalo gourd, which is really very prevalent in our growing region here, we do see a lot of them symptomatic. And when we sample and test for viruses, we do find different virus combinations in them. I must mention that there are some of these wild cucurbits, like smell melon, that actually can harbor some of these infections without showing symptoms. And so, it's really very important to sample, some of these wild hosts, especially they are very, when you find them very close to your growing field. So in terms of the host range, relative to some other begomoviruses like tomato yellow leaf curl, for instance, it's fair to say that the host range of squash leaf curl virus is fairly narrow. It's primarily in the cucurbits, the ones I've mentioned, pumpkins, cucumber, squash, watermelon, melons. But also wild relatives of those cucurbits. But some strains of squash leaf curl virus are known to also expand their host range to include plants in different families. However, the precise roles of these non-cucurbit hosts in disease epidemiology is really unclear. In other words, we are not quite sure whether they can be efficient sources of infection of cucurbit crop. So that's something that really needs to be elucidated. In terms of the distribution, every yellow dot you see there shows a country where squash leaf curl has been identified. And these countries, they span Asia to South America, to Central America, to the Caribbean, but also, of course, to the United States. And as far as distribution in the United States is concerned, like I mentioned previously, it was first observed in California in the late 1970s, and about the same time, about a year after in Arizona. But subsequently, it's been identified and characterized in Texas in the early 1990s. And, there is a recent, paper from 2012 that shows that it's also present in Mississippi and Alabama. Strangely, we have no report of squash leaf curl virus present in Georgia and Florida, 2 other major cucurbit producing states. The transmission is going to be very similar to what you've had. It's transmitted by the silver leaf whiteflies in a persistent circulative manner. It is not, it doesn't pass through the eggs, so it's not transovarially transmitted, and it's also not seed transmitted. Just like Sudip mentioned before, once the whitefly acquires the virus, squash leaf curl virus, it actually retains the ability to, harbor that virus for life and transmit it. Within an across field spread, of course, it's aided by whiteflies, and we have to keep in mind, that when those whiteflies are aided, by wind or even things like hurricane currents, they can actually move over across, geographical boundaries, very far. And like you've heard before, also untested transplants, especially as people use transplants, are things that could also be vehicles for long distance spread, especially when those transplants are not yet showing symptoms of infection. But also something to keep in mind is that, some of these whiteflies can acquire the virus and infest ornamental plants that are traded more frequently. And those can also be vehicles for long distance spread. So it is very important not to inspect these transplants as well as ornamentals, not just for symptoms of virus, but also for indications of a whitefly infestation. In terms of diagnosis, symptoms-based diagnosis is definitely unreliable for the reasons I've advanced previously and spoken about by other speakers. Number one, there's a lot of overlap in symptomology of these different viruses, but also the fact that mixed infection seems to be the rule rather than the exception. And so it's difficult to look at a plant and just, based on symptoms, say it's for sure just single infection or squash leaf curl. And so you really have to take a sample, send it to a lab that is equipped to test for squash leaf curl and other cucurbit viruses. And there are different methods that you've heard about that can be used, serological methods, but also field deployable methods like lamp assay. I must emphasize here that some of these are really not readily accessible. And so most diagnostic labs, still rely on PCR-based diagnosis. There are several published primers and protocols, some of which you'll find on website, the Emerging Cucurbit Working Group website. And some of these have been published, they've been peer reviewed, and they are available for use. Something also to mention here is that, even when you have very low titers of the virus within the plant, you can actually enrich, that template, through this rolling cycle amplification step to improve the detection signal. But something I would like to mention here is that even with PCR-based diagnosis, there's always a chance of some of these primers cross-reacting or amplifying other begomoviruses that may be present in mixed infection. And so, especially for new set of samples, it's always not a bad idea to consider sequencing the amplified DNA fragment, to be very sure that what you've actually amplified is squash leaf curl virus. In terms of management, it's going to be similar to everything you've had. Host resistance, of course, is desirable, and there are some sources of resistance that's been identified in some cucurbita species, but many of those have not really been incorporated into commercial varieties. But there are some zucchini varieties with intermediate levels of resistance. Avoiding overlapping cucurbit crop production is really very important so that we don't have continuous green breach, for squash leaf curl virus and other viruses. And of course, because wild cucurbits, harbor this virus and they could be sources of infection, it's also very important to make sure that we are paying attention to them and roguing them whenever they are present within or around the field. Use of green transplant is something you've heard about rather than direct seeding, because, when the plant gets infected at very young age, the potential for severe losses is higher. And so transplants, can help, to alleviate that. And especially if you can afford, to use row covers for those transplants, that's even something that is really very desirable. Something to keep in mind with using row covers is, you really have to be on top of your game in managing some foliar fungal diseases because, some of those row covers provide a perfect condition, for some of these fungal diseases to really, take off. So it's really important to keep that in mind. Making sure that we're on top of whitefly management, either using chemicals or physical barriers, like I mentioned, is really going to be very important. Scouting at the field early is something I always recommend to our growers here. And when you see a plant that looks suspicious with virus-like symptom, when in doubt, it's probably not a bad idea to take a sample for diagnosis and sacrifice that plant so that you remove or eliminate source of inoculum. But it's very important to mention here that while each of these methods are really effective, using them in combination in a proper IPM program is going to be more, more effective, than using them singly. So with that, I would like to thank all those who have contributed to some of the work we've been doing in the past and current students and postdocs, growers who have allowed us access to their fields and industry cooperators, my research collaborators, as well as those who have funded some of the work we've done.
All right. Thank you, Femi.